this evening with you, Jen, especially, and celebrate all that we've accomplished. Why don't you tell us about what we've done?
Well, we have done quite a bit these past 11 years. We started the foundation in 2010. I'd love to give you a little bit of history about how we got started, how Missy and I got connected all those years ago. Unfortunately, we missed our real big celebration last year. The pandemic put our 10-year anniversary sort of in the back seat. So this is sort of a little pseudo 10-year plus one anniversary celebration. So I'm glad everyone is able to join us. Missy and I go back a long way. I reached out to Missy in 2007, right after my daughter was diagnosed with this rare syndrome. And Missy and I became fast and furious friends right from the start. We had quite a bit in common, but I would say the biggest thing we had in common was this desire to find some answers and to get some hope for the situation that we both found ourselves in. So in 2010, we decided to form the SMS Research Foundation. We felt we needed a place that was solely dedicated to funding research. There were several different organizations going on at that time, some designated to awareness, to services, to whatever kinds of other things you needed, but nothing that was 100% dedicated to funding research. We came together, and in that first year, I actually was going back and looking it over. We did 4 events in that very first year. And within, I would say, less than 11 months, we funded our first research grant of $50,000 to Dr. Lubsky at the Baylor College of Medicine. And after that, we just took off from there. Along the way, lots of families joined us, more events, all different kinds of things we did, from marathons to triathlons, to golfing events, to whatever we could think of. And so many of you out there joined us to get to where we are today, which I am so excited to share is over $1 million dedicated to research. Later tonight, you're going to hear all about the projects we've got funded right now. You'll hear from the head of our SAP, which will be very exciting. And our researchers will also join us later, which really is the first time ever we've had an opportunity to do that. I would like to just give you a little bit of history about the foundation. Again, Missy and I, really with no experience whatsoever, started this organization, the quintessential grassroots mom and pop type of a beginning. And we have built quite a successful organization. We've had numerous board members, and I'd like to tell you a little bit about what we are built upon, our core values, which is what drives our foundation. We have, here's a slide coming up so I can show you our core values that we really worked hard to put together. Very important items for us that we really hold to when we work towards our goals: accountability, curiosity, integrity, trust and candor, humility, commitment to excellence, and empathy. All very important, and that really is the core of our organization. I'm going to hand it over to you, Missy, so you can introduce all the people who have joined us along the way.
Thank you, Jen. As they say, it takes a village, and it has certainly taken a village to get this organization up and running. I could not be prouder of the team of board members that we currently have on board. We are spread out all across the country, but we managed to get the work done, and we work amazingly well together. So I'd like to introduce every one of our board members individually, beginning with Jen and Chris Ianuzzi from Connecticut.
And Missy and
Oops.
Hey everyone, my name is Ianuzzi, and I'm one of the co-founders and CFO of the Sms Research Foundation. And I'm here with my daughter, Sienna.
Hey, everybody.
So we just quickly wanted to talk about why research is so important. When you have a complex and very rare syndrome like SMS, there's very little funding that goes into it. So we felt obligated over 10 years ago to form the SMS Research Foundation. And our hope and dream is that someday through research, we can find some viable options that will improve the life of these children and adults with SMS. So that is our goal and that is our mission. With that being said, you want to say goodbye to everybody? Bye, everybody. All right, thank you all. Take care. Bye-bye.
Thank you.
Next, we have Paul Diamond from California.
Hi, my name is Paul Diamond, and I've been a board member at the SMSRF for about 8 years now. Our son was diagnosed 14 years ago. Of course, probably the most impactful thing to happen in our lives is his diagnosis of having SMS. And our way to give back, to fight back in some ways, is to support research, because research will help improve the lives of families that are being diagnosed today and out into the future. Finding that treatment will make all the difference, and research is the path to get there. So thank you so much to everyone who has donated and supported this great event and the foundation in general. Thank you.
Michael Kingston from California.
Hi, I'm Ben.
And I'm Mike, Ben's dad, and also a board member with the SMSRF. When Ben was first diagnosed with SMS, what we realized is there's quite a bit known about the characteristics of individuals with SMS, but little known about the disorder itself, what causes it. There certainly aren't any therapeutics available for individuals with the disorder. And so we were thrilled when we found the foundation and all the wonderful research they're doing and the fantastic researchers they collaborate with, and that's why I joined the board. And we're really excited this month by the walk, and we're really thankful to everyone out there that donated. So just a big thanks from both of us for all the donations and all the great walking that everybody did.
Bye.
Manny Garsinski from Wisconsin.
Thank you all for being here. Why did I join the Mcginnis Research Foundation board? When we got through those diagnoses, I did a lot of reading and research about what was being done to help those with this rare syndrome. The SMS Research Foundation had the most exciting basic research taking place. We have 3 great researchers who are currently tackling various aspects of the syndrome, and we're hoping that there will be some therapeutics and treatments in the near future that could help these kids and adults who suffer with SMS. Thanks for supporting these efforts. I just got in from a walk right now, so thanks again for supporting me and supporting this great organization.
And Jonathan Miller from California.
Hi, I'm John Miller, and this is my son, Griffin. Can you say hi? Hi! Griffin got diagnosed with SMS when he was about 3 weeks old, and over the years, we've looked for ways to try to be involved in helping Griffin and other kids like him. And I realized that spending time with the SMS Research Foundation as a board member is the best way to do that. And I'm really excited about all the things that we're trying to accomplish and grants that we're trying to fund, and look forward to continuing to help the organization going forward. You say bye-bye!
We would also like to recognize some of our past board members who have joined us over the years. Michelle Zandowski, Nicole Gerberg, Monica Mason, Amanda Serio, Susan Diamond, Steve Woods, Jessica Rogers, Laura Staich, and Courtney Frist. Thank you so much to everyone who has dedicated their time to helping us grow. At this time, we are going to turn my seat over to the head of our scientific advisory panel, Christopher Ianuzzi. He is going to share with you all things research.
Thanks, Jen. Appreciate it. So my name is Chris Ianuzzi, and I am the chair of the Scientific Advisory Panel, and it's really great to be with you this evening. I'm really excited about all the research that's going on, and really excited to share our process and to talk with our researchers tonight about the progress that's being made. So before we get started with that, though, we'd like to show you a short video So, we recognize that it is difficult sometimes to explain to even friends and family what SMS is, the genetic sort of changes that take place, the implication of those changes, and some of the research going on. So we put together a video that simplifies that process, and we hope that you'll use it to communicate with friends and family. You'll be able to get this video on our website and on our social media feeds. So Terrell, please, if you can show the video.
Smith-Mcginnis syndrome, or SMS, is a complex genetic disorder. It is referred to as a microdeletion syndrome because a small part of DNA is either missing or not functioning properly. Every cell in the human body contains 46 chromosomes, 23 pairs. A chromosome is a long piece of DNA that contains the genetic information of an individual. This genetic information is sectioned into genes that are unique sequences derived from parents and transferred to offspring. Genes code for important proteins that play a critical role in human cell regulation, facilitate proper growth and development, and give us our unique characteristics. For those with SMS, one copy of the RAI1 or retinoic acid-induced 1 gene is missing, or one copy is mutated, such that it is rendered non-functional. Therefore, this gene is not functioning at the proper level. This has detrimental effects on all the proteins in the cell that it would normally interact with to do its job. The brain is a key tissue that requires proper RAI1 protein levels for growth and development. The lack of sufficient RAI1 production leads to significant clinical and behavioral issues, which can include self-injury, aggression, impulsivity, emotional dysregulation, obesity, sleep disturbances, and cognitive impairment. Unfortunately, this constellation of features makes it extremely challenging for individuals with SMS to become fully independent, and they typically require a lifetime of supportive care. The SMS Research Foundation is dedicated to finding unique and innovative ways to improve the lives of individuals with Smith-Mcginnis syndrome. Our research is based on 3 approaches. 1, replace the bad copy of the RAI1 gene with a good copy using gene therapy. 2, stimulate overproduction in the good copy to make up for the bad copy using Crispr technology. 3, find or develop a drug that can promote the production of RAI1. If RAI1 can be replaced and properly regulated, it is possible that some of the most difficult features and cognitive delays may be improved. Supporting this kind of research will lead to the most effective treatments for people with Smith-Mcginnis syndrome.
Perfect. Thank you, Carol. So now if only it was that easy. And so now we'll talk a little bit about our research process. So if you can show the next slide for me, Terrell. So our scientific advisory panel, we believe that strong science starts with strong people. And so we have put together what we think is a very impressive group of individuals to help us determine what projects should be funded and how to allocate those funds. So our scientific advisory panel is made up of people that represent basic science, people who run their own labs and understand the true costs that are associated with these projects. We have people that are associated with clinical medicine, people who understand the clinical aspects of the syndrome. And then we have people representing the pharmaceutical industry, people who understand drug development and the steps necessary to develop new and novel therapies. And it is this combination of people that allows us to be good stewards of the money that you entrust with us. And so our scientific advisory panel is made up of Gianluca Perazzi. You're going to hear from him a little later this evening. He's a senior vice president and clinical development head at Alexon Pharmaceuticals and also a father of a child with SMS. Li-Chin Liu, who's a full professor of biology at Stanford and lead investigator at the Howard Hughes Institute. Luann Hudgens, who's a full professor of pediatrics and medical genetics at Stanford University. Sang Cheng, who's the senior vice president and chief scientific officer of rare diseases for the Pfizer Corporation. And then Weimin Zhang, who's an associate professor of molecular, cellular, and developmental biology at Yale. Next slide. So it's those folks that help us really outline our strategic research vision and set our priorities. And we have 4 basic priorities that we try to hold true, and we're deciding what research projects that we're going to fund. Number 1, we want to gain increased understanding of RAI1 and its role in normal brain development. We want to reestablish normal cellular RAI1 expression. In other words, We want to fix what's broken. We want to try to put back the function of RAI1. We want to study the neurobiology of cognitive and maladaptive behaviors. And we want to develop new and novel therapeutic approaches to SMS. And we encourage innovation. Next slide. So we've tried to standardize how we allocate those funds. And where we used to fund research, once we had the appropriate funds in place, we've actually now gone to more of a grant cycle where we try to fund a new project every year. And this we find is a way to allow to give seed money to new investigators that want to start to involve themselves in SMS research and RAI1 research. So it's generally $75,000 plus $6,000 for what's called indirect costs that go to the academic institution. So it's a total of $81,000 per year. And generally, a lab would first submit a concept paper, which would allow us to get an understanding of what the proposal was. And if we felt that was in line with our mission and our priorities, then we would ask for a full proposal. And then we use an NIH scaling format to rate each project, and then the highest rated project would be funded. And so we've been doing this grant cycle for the last several years. And if the project is particularly promising, then after that first year, we have the ability to extend that project. And we'll be announcing later this evening that we've done that in the past, and we're going to do that again. Next slide. So right now, we're actually funding 3 labs. We're funding Dr. Sarah Elsie at the Baylor College of Medicine. We're funding Dr. Wei-Sheng Wang at McGill University in Canada. And we are funding Dr. Michael Higley and Dr. Jessica Carden at Yale University. And so now what, and if you have any, and although there's not enough time for questions this evening, I would like you to know that if you, after you hear from the researchers, if you do have questions, you could reach out to me personally at cmianuzzi@msn.com. Our contact information is also on our website, smsresearchfoundation.org. And you can also ask us questions directly, and we will make sure that we get back to you. So now I'm going to show you a video from Dr. Perazzi, Gianluca Perazzi. I think most of the folks in the community know him. He's the father of a child with SMS. He's a physician. He's a PhD scientist. He works in the pharmaceutical industry. You couldn't build a better person that can be, that should be part of our scientific advisory board. And so, although he couldn't be with us tonight, he put together a video to show his enthusiasm about the projects that we're funding. And I think we'll show that video next, Terrell.
Good evening, everybody. My name is Gianluca Perazzi. I'm the father of Valentina, a 7-year-old girl with Mcginnis syndrome. I'm also a medical doctor, and I work in the pharmaceutical industry. In fact, my job is in research and development, researching new medication for rare diseases, just like SMS. When my daughter was diagnosed with SMS, I did what many parents do. I Googled SMS. In fact, despite being a medical doctor, I never heard of SMS before, and that's often the case with rare diseases. As a parent, learning about SMS made me confused, exhausted, and distressed. As a medical trained in pharmaceutical research, learning about SMS made me curious. At that time, there was minimal or very little in general research on SMS worldwide. So I saw the opportunity for a dedicated research and collaboration, and honestly, the responsibility to do something, the responsibility to try to develop a medication or any research focused on looking for therapeutic intervention and medication that could help our children. Soon I connected with the Sms Research Foundation. And over the past few years, our Sms Research Foundation Scientific Advisory Panel, I'm part of that, has grown to include scientific and clinical experts specializing in rare diseases and genetics. Together, we direct and supervise the scientific efforts of Sms Research Foundation. Now, starting new research is difficult. There's very little known about SMS. Research is expensive and is time-consuming. But we're able to launch 3 programs. So our starting point, which was really the roadmap of our vision as an SMS Research Foundation, was to tackle the disease from the very beginning, from the core. Now, SMS is characterized by a haploinsufficiency of the gene RNI1. What does it mean? It means that one of the two genes is missing due to the microdeletion of the chromosome 17. In this situation, there's multiple ways to try to replace the gene or the missing function. So we tackle the problem by using multiple approaches. So we say multiple shots on goals. That's why we have 3 programs currently ongoing, financed by the SMS Research Foundation and by your efforts and your donations. The first program is gene therapy. What does it mean? Well, basically, one way to replace the missing gene is to use these new technologies that allow to put the gene back into the body, into a human being. And we're focusing, of course, on the central nervous system. Now, when you start the gene therapy program, you always start developing an animal model and trying to replace the gene in the animal model. And if it works in the animal, you then move to human. We've been working on this for a very long time, over 4 years. In fact, this is a very complex approach that requires many efforts. And after developing several vectors and trying several approaches, we now finally found one that works, which includes the use of Crispr-Cas9 technology. You probably heard about this technology. It's a fairly new but famous approach now. And we have data in an animal model that shows promising results, which are the base for NIH application for a grant. This work has been run by Sarah Elsie, Professor Sarah Elsie from the Baylor College, and you will hear more directly from Sarah today. The second approach is different. The second approach is run by Professor Wei-Jian Han from McGill University in Canada, and he's looking at increasing the activity of the protein produced by the gene. So what it means, because there's one gene missing, the protein produced is less. Now, if I can stabilize the RA1 protein or increase its expression or increase its function, I can basically replace the missing activity and somewhat improve the symptomatology of the disease. So as you can see, this is a very different approach as compared to gene therapy. And the type of therapeutic that could stabilize the protein is generally given as a small molecule, which basically translates into a pill that will have to be given every day. So we are at the very beginning of this, looking at potential medication that can stabilize RNI1. And obviously there are multiple efforts to be able to find potential options to do that. The third program we're working on is I would say, fairly different scope. The scope is to understand the underlying physiopathological mechanism of the disease, the origins of SMS, and potentially work on therapeutic understanding targets for therapeutic intervention, and also ways to measure response to treatment in SMS. This program is run by Professor Mike Higley at Yale, and you will hear more about this as well. This is the latest of our program, and we're very excited about advancing knowledge and understanding from physiopathology, with the goal, again, to develop potential therapeutic intervention. I'm incredibly excited about all of this research. I hope you understand and you see that this is pretty cutting edge and is very advanced and very exciting. And I would like to thank everybody on the call for your support and for your direct involvement in the SMS community. This community helps raise awareness of SMS across an academic institution, members of healthcare community, other families who are affected by rare diseases. So let me thank you to everyone for watching this video. Thank you for the efforts that you do to support our research. And I'm confident we will continue working together, achieving great things moving forward. And now, let's hear from some of our researchers about the incredible work they're focused on. Thank you.
So now we'd like to hear from our researchers, and the first researcher we'd like to speak with is Dr. Sarah Elsie, who's a full professor of molecular and human genetics at the Baylor College of Medicine. It's really been a pleasure and my honor to have been working with Sarah for the last several years. And so she's really been the head of our signature gene therapy project. She's going to give us an update this evening. And so welcome, Sarah. Thank you so much for taking the time to speak to us. And you can share your screen if you don't mind.
Great. All right. Can everybody hear me?
Yes, thank you.
Okay, great. Thank you, Chris. Yeah, really happy to have a few minutes to tell you about the work that we've been doing and some of the exciting progress that we've had over the last few years. And so as Gianluca and Chris have explained, some of our goals really have been to try to improve and increase expression of REI1. That's really been one of our primary goals. The last year, we've had some challenges. Of course, everybody, with the coronavirus challenges, we've had to shut down our laboratory and stop breeding mice and growing cells. And so we've had some challenges along the way. But we've, I think kind of overcome some of those and kind of powered through. And I think we're back up and running and in good shape. The philosophy we have in my lab really is that we have our research, everything that we do is really patient driven, and all of the findings and such really have been driven by information we've received from parents and from patients, and to drive the focus and the goals in the research to really try to get to the improved quality of life, right? That's really our goal in the end. And as scientists, I think, having that communication is incredibly important, and to make sure that what we're doing actually can be meaningful and have a meaningful translation to the patient in the end. And so that is certainly our goal. And I don't have time to tell you tonight about all of the work that we're doing, but some of you may have provided or children may have provided skin biopsies for research purposes. And I won't talk to you tonight about that work, but too much, but we do work with those as well. And so we've created stem cells and neural stem cells to further assess REI1 and to determine kind of the limits, basically, of what can we do with replacement of REI1 or increased expression of REI1, and to really fully assess this. So we're looking at cellular and whole organism level. So one of the greatest challenges in doing this work is that REI1 is what we call a dosage-sensitive gene. And so you may know that as you know, not enough REI1 gives us SMS, but too much REI1 gives us Potocki-Lupski syndrome. And so we do want to make sure that we balance this and that we really keep dosage within a reasonable level, so that we do not create a problem with the treatment that we are doing. And so that's one of our challenges. And so our primary goals, as Gianluca and Chris I think really highlighted, is that we're looking at function of REI1. Everything that we do not only works toward a treatment and then the treatment goal, but also we learn more about REI1 along the way. And so that's been very important process for us, and trying to understand those outcomes in the patient, and how can we modify those outcomes in the end. And so, as I mentioned, genetic approaches, drug screening approaches, and various clinical trials that we're doing as well that are outside the funding of this particular project. And so what does REI1 do? Well, we know, as you know from your children, that REI1 is involved in function of circadian rhythm. It's involved in development, metabolic function, and neurological function. And so we're trying to understand, can we impact any or all of these aspects of the condition with a therapeutic approach? And
Sarah,
so that...
I apologize.
Yeah.
We don't see your screen. Are we supposed to see your screen?
Yeah, you should be. Oh my goodness, you're not seeing it. Oh, I'm so sorry.
It's okay.
Gosh.
We're seeing you.
So I'm just chatting and you're not seeing anything. I apologize.
So if you
Let me pull it up. Let me share. Let me see if I can share it again and try. I thought I was sharing. I apologize. Let's try it again.
interrupt you, but I thought you were
Let's
trying.
see. It's embarrassing. Okay, let's see. Let's try it again. Is it coming up now?
Yes.
Is that coming up now?
Yes, perfect.
Great. Okay. All right. I apologize for that. So, so as I mentioned, we're really trying to understand, can we impact these aspects of the condition by increasing or providing additional copies of REI1. And this just gives an idea across the cell. When you have haploinsufficiency of REI1, there are many different pathways that are affected. And so while we see behavior and obesity and circadian rhythm abnormalities, all of these pathways do not work in isolation. So they do all work together. And so we want to be cognizant of all of those potential impacts. And so we use a variety of different model systems to study SMS and over the years, and from a variety of different animal models to cells and for these projects that we talk about today, we're going to primarily focus on mouse. And as I mentioned, we also are doing cell-based work, but the primary focus will be the mouse work. And of course, our therapeutic goal overall is the increased expression of REI1. And that's really our primary focus. And you can do that by increasing expression from the non-deleted chromosome 17. That's one option. Or you could introduce another copy of REI1 into the cell. And we've attempted and trying to do both, taking both approaches. And can we alter REI1 and expression in the right cells? Because that's important, because not every cell expresses REI1 at the same level. And so we want to make sure that we try to get it to the right cells at the right level of expression. And again, how can we best measure success in this treatment process? And so lots of different approaches that we've taken over the years, and with regard to trying to replace the entire REI1 gene, which is challenging because it's a very large gene physically, and so it is difficult to fit into a gene therapy vector. But we have created cell lines that have corrected the expression of REI1 and in the stem cell form, and so we do believe that we've got some great approaches along those lines, as well as the approaches that Gianluca had mentioned with regard to a CRISPR Cas9 approach. And so this CRISPR approach is illustrated here briefly, that basically what we're trying to do is to introduce a deactivated CRISPR. So this is not a CRISPR that will cut the chromosome and make a change in the DNA. What this does is brings cofactors to the DNA and helps increase the expression of REI1 from that remaining chromosome that is not deleted. And so that really is our goal. And so we've had some good success with this. And so, as I mentioned, how do we measure the success? Well, that's always a challenge, I think. And this illustrates, I'm going to show you a little bit of data on mice that are not treated. So these are SMS mice that, and you can see the weight gain here. The red and green are wild type animals, okay? And so there's a wild type female on the bottom here and male animals. And then the black and blue illustrated are male and female REI1 mice. And so you can see that these mice, while they start out relatively close in size, as they age, they really become quite obese. And these are quite large mice. And so this is one of the primary phenotypes that we're monitoring as well. And so the mice weigh a lot. They have altered fat deposition. They also have impaired satiety. And so they tend to overeat as well. And so one of the mechanisms by which we do this monitoring is using a metabolic cage. And it's a little hard to see some of these because we don't have fantastic cameras to put inside near the cages. But this guy in white is a Smith-Mcginnis mouse, and he's trying to run on the wheel, and he's not quite keeping up. It's a little hard to see this guy here, but this guy's running. This is a wild type sibling of his who is really running hard on the wheel, and this guy's a little challenged. He's quite overweight, and you'll see him kind of give up here in a minute. And his siblings are running all the time. Okay, and so we use this measure of activity and weight and food consumption and energy expenditure as part of our analysis to determine if we've had some success. And so mice that we have not treated are illustrated here, and this is food consumption over a period of time. So this is total food consumption over a period of time while these mice are in the metabolic cages. And in the black and blue illustrated are the REI1 mice, and so these are females in black and the males in blue. And you can see that they eat over time a lot more food than the wild type animals, which are shown in blue and red and green. Okay, and so this is typical. We've seen this in over the years, and we know that, and we expect that this is what we will see in these animals. And shoot, it's not advancing. Okay, and so this is the wheel running, and as I'd shown you there, you can see that the animals they're just not very active. The red and the green here illustrate again the wild type animals, and so you can see they're running, they're all over the place. The female mice are really active, more so than the males, but the Smith-Mcginnis mice are shown down here in the blue and the black, and they're just not that active. The males are a little more active than the females, but the females are saying, thinking, I am not getting in that wheel. I'm not going to run. So they're really not very active. Okay. And so when we do injections, so those are mice that have not been treated, right? So those are just animals that, and a typical phenotype. And so in the animals that we're treating, we're injecting an AAV2 gene therapy vector into newborn pups. And so this is the day of birth for these animals. This is called P0, and this is the day of birth. So these animals get injections into each hemisphere of the brain, and with the idea that this will very readily spread throughout different sections of the brain and go readily into the cells. So we do one injection and then put the mice back in with mom and let them grow. And so then we do a first analysis at 6 weeks of age in the metabolic cages. And you can see here that in we have saline injected, which is a control animal. And so those are in pink and blue. Okay, so you can see the pink is the heterozygous, which is the SMS mouse. And so you can see this SMS mouse, this is food consumed. You can see it goes up there pretty high. So that's the heterozygous. And then there's the AAV injected. And you can see that it's pretty similar, right? And in fact, the wild type AAV injected also has increased food consumption. So the food consumption is not so dramatically altered in these animals, and which was surprising to us. But let me move here. So food intake minimally impacted. Okay. However, as we go on and look at other different aspects of the phenotype, what we find is that when we look at activity assessments over time, we find that animals that are saline injected, which are shown in pink, not very active, as I had shown you, right? However, when we inject the heterozygous animals here in black, they have now the same activity level as their wild type litter mates. And so this is really fantastic. And so we're very excited about these data. And the wild type injected animals, these replicate Potocki-Lupski syndrome. And you can see that the activity is much higher in those animals. So we don't want to get there. We want to keep them here where we are right now. And this looks pretty good. And so we're quite excited about this. So what we know is that treatment improves activity and energy expenditure at this age. It's not altering the satiety impact, and we need to do more studies to understand why that aspect is not being altered substantially, but the energy and activity is, and so that's very exciting. And then when we look more so at not just the energy expenditure, this is just another representation of energy expenditure from control untreated animals. But what we find is that this energy expenditure also correlates, as you can imagine, with your circadian rhythm as well. And so we do see shifts that occur related to the circadian rhythm, and it appears that with the treatment, the animals are shifted to a more normal circadian rhythm, like their litter mates. And so we're very excited about that aspect as well. And which, of course, as everybody knows, that would be a huge improvement if you could improve the circadian rhythm. And so in this, as I mentioned, the dosage sensitivity is challenging, and that's something that we're constantly trying to work with and understand, as well as making sure that we get this treatment into the right cells at the right time. And so that's where our research is going now. And as you can see, we've got some really great data, but we have much more to do. And so, as you know, it's the outcomes of this kind of research are really fantastic, because not only does it help Smith-Mcginnis syndrome, but it helps the broader population. And that's important for all of us to try to put out there as well, is that if we can understand this single gene disorder that impacts sleep disturbance and obesity and behavior, that then those are also common problems in the population. And so this also helps us understand treatments for others as well. So that's very important. And lots of people in my group that have been involved over the years in this project and related projects. And of course, I couldn't do any of that without them. And of course, with the funding, that's incredibly important. And my other collaborators from other institutions as well. So I'm happy to take any questions.
This is great, Sarah. Thank you so much. We really appreciate it. And very exciting data. I mean, the fact that you can, increase expression of RAI1 with that CRISPR technology and start to see some normalization is really exciting. So we recognize that this kind of research takes longer than we expect, and it's more expensive than we expect. So I think it's also exciting to know that you put in for an NIH grant. So hopefully that can bring additional funding and additional awareness to SMS and to RAI1 research. So we're proud of that, and we thank you for your collaboration and all that you do for children with SMS.
Great.
Thank you very much. I have a short video. So Dr. Wong from McGill couldn't join us this evening, but he put together about a 5-minute video to talk about the life cycle of RAI1. And his approach, as Gianluca was stating, is a different approach than Sarah. He's looking more at a small molecule approach, looking to try to decrease the degradation of RAI1 so that it hangs around in the cell longer. And so he'll talk a little bit about that next. And we use subtitles because the sound is not perfect.
My name is Michelle Huang. I'm a professor at McGill University in Canada. I did my PhD at Baylor College of Medicine in Texas with Dr. Ianuzzi. She is a pioneer in child neurodevelopmental disorders. Then I moved to California and did my postdoc with Dr. Li-Chun Lo at Stanford University. At Stanford, I became interested in Smith-Mcginnis syndrome and the molecular as well as neurobiology of RAI1. At Stanford, I discovered that if you increase RAI1 level back to normal, you are able to rescue some disease feature in a mouse model of Smith-Mcginnis syndrome. We also did different deletion to find out which brain regions depends on RAI1 for normal function. So building on these exciting findings, in my own lab at McGill, we are interested in studying the molecular function of RAI1 and hoping to find a cure for SMS. Today, I want to illustrate some of the ideas that we have for finding the cure for SMS. So first of all, I want to talk about the life cycle of RAI1. RAI1 was induced during development by a retinoic acid molecule. So it binds to the receptor, which translocates into the nucleus of the cell here to activate RAI1 gene expression. After RAI1 is produced, it will be released into the cytoplasm here, and it will be modified by different chemical molecules. Some chemical molecules will make RAI1 more stable, some will make them RAI1 less stable, and some will basically target RAI1 for degradation. And RAI1 will come back to the nucleus and binds to the DNA and regulate gene expression. And we know the function of these RAI1 downstream target gene is very important, because if you have less RAI1, the expression of some of the genes will be suboptimal. And this is why we think it will cause disease. And knowing this, we decided to pursue 2 different strategies to increase RAI1 level or target downstream RAI1 downstream molecules. The first strategy was by identifying the different enzymes that modify RIL1 stability. We are focusing on different kinases and phosphatases because those molecules are commonly targeted by drugs. We hope to identify those molecules and use drug to inhibit those molecules to increase RIL1 stability. Our recent finding funded by SMS Foundation showed that RIL1 is highly unstable in the cells. So by stabilizing RIL1 in the cells, we hope to increase the level of RIL1 and hope we can improve the symptoms of SMS. Another strategy was to identify important RIL1 downstream targets. We know there are many different RIL1 downstream targets, so we are trying to dissect each one of them to see which one is the most important one. And hopefully we can focus on the more druggable molecules and target them for therapy in different animal models of SMS. I want to emphasize that all of these discoveries and ongoing works they were supported by SMS Research Foundation. Without the help of the foundation, it is not possible. I would also like to thank all the family for support SMS Research Foundation. Although research is difficult and challenging sometimes, I promise we will not stop finding the cure until we find one. Thank you.
Great. So now, for our finale, we'd like to speak with Dr. Michael Higley. Michael is a professor at the Kavli Institute for Neurosciences at Yale. And Mike's a great example of our annual grant cycle. So he received a grant a year ago for what's called cortical dynamics, which he'll discuss in a little more detail. And then we are really proud to announce that actually our SAP was so impressed with his research that we've decided to fund him for an additional year. And he'll talk about what he's going to try to accomplish over the next year. Mike, are you with us?
Yeah, so look, I'd first just like to say thank you so much for having this evening and giving me a chance to tell you guys, sort of what we've been up to and at least get to communicate a little bit of our excitement and what we're up to the families that have just been so incredibly generous to support us in this work. So what we are primarily engaged in is trying to understand or phenotype, as it were, the kinds of brain activities that are associated with dysfunction of this gene RI1 in mice. And so, as Chris says, my name is Mike Higley. I'm an MD PhD in the Department of Neuroscience at Yale University. And this work is done in close collaboration with Jess Carden's lab, also in the Department of Neuroscience as well. And, one of the things that Chris asked us to do is sort of let you know how we came to this project and really got involved with the community. And quite honestly, the way it worked for us is that we're a lab that primarily has worked in understanding basic brain function using mouse models, and an opportunity to sort of apply some of our work to a number of models of neurodevelopmental disorders came along. And we also saw then this opportunity from the SMS Research Foundation, and we applied with some of our best ideas, and we're incredibly fortunate and thankful to be funded. And it really started us on this journey, which is turning out to be incredibly interesting for us, and I hope valuable to the community and the research efforts directed at RAI1 and understanding some of the mechanisms underlying Smith-Mcginnis syndrome. So just to give you a little, and so again, so this is a project that was generously funded by the Research Foundation, and so as you've heard, quite a bit, Smith-Mcginnis syndrome ultimately derives from dysfunction or loss of function of the gene RAI1. And there's a lot of efforts, both supported by the SMS Research Foundation, supported by NIH efforts, by other foundational efforts as well, to look at cell signaling, how individual cells, especially brain cells called neurons, may work or not work, how the synaptic connections, the chemical connections between neurons might be disrupted, how that affects the activity of those cells, how the wiring, essentially the circuit map of brains, might be altered when RAI1 is not doing what it's supposed to do, and also how sort of the whole circuit works, how cells interact together And what we think is that behavior is ultimately the product of these complex interactions of different cells and cell networks. And in fact, that's where our work has really come in. So there's a lot of amazing work, some of which you've heard from over just the last few minutes, at more cellular and genetic levels of analysis. And what we've been trying to tackle is a little bit from the other end, trying to understand how gene dysfunction leads to altered brain activity and how that might contribute to some of the behavioral changes that we see in SMS. So our model is to work with mice. It's fairly common. And one of the things that I'm incredibly thankful for to work with this community in particular is it is a completely reasonable question to ask why we use mice to model such a complex disorder that is clearly affecting, human patients, human kids. And it's a good question. And the truth is that there are many experimental methods that we can use in mouse models that are not accessible in humans. And so we would consider this what's called a preclinical model, which was we are using the mice to understand both basic brain mechanisms and processes that go wrong when RAI1 is disrupted, but also, and I'll touch on this very briefly in a moment as well, to use it as a test bed for understanding or for potentially vetting new therapeutic options. The idea being if we see something wrong in a mouse and we develop treatments that sort of correct that in the mice, there's at least some reason to be optimistic that those methods might ultimately provide some benefit to the human patients as well. And so we use mice in which one of the copies of RAI1 is disrupted, is missing. And we use viral vectors, in one case, something called an adeno-associated virus, to express a green fluorescent indicator in the brains of these mice. So this is a protein that isn't there normally. We put it in with a virus, and when their brain cells are active, this protein lights up. And we could use fancy microscopes to use that lighting up to look at how brain activity is unfolding in sort of the awake behaving mouse, and also compare and contrast what we call wild type mice with mice that may be missing a copy of RAI1. And so we have recently developed a set of microscopic approaches, one of which is called, we call it Mesoscopic Imaging. And this is sort of a fancy word to mean sort of middle. So we are not looking at single cells, which we might refer to as microscopic. And we're not looking at, say, the whole brain as you might with functional magnetic resonance or fMRI, but we're looking at sort of a big picture of one big circuit, the neocortex, which in humans is the big part on top. And so we can look at the entire neocortex in an awake behaving mouse at one time and assess the dynamics of its activity. And I'm going to show you just a little bit of a movie that's just kind of fun to see what some of this looks like. And so hopefully you can all see this play. And so this is essentially a top-down view of a mouse brain. This is the front up, the back, and then in terms of the left and the right, you can actually see some of the blood vessels, these lines going across. And these flashing patterns of activity, I think I can play it again. Everywhere that it's sort of dark blue is quiet. Everywhere that it gets really bright yellow or red is an area that's active. And what I hope you can appreciate is that this is a mouse just sort of sitting there, hanging out, and the brain is incredibly active. There are patterns of activity. Some regions are active together, some are in opposition. These patterns change over time, depending on what the mouse is doing. This is essentially a real-time readout of how the brain is functioning and potentially how it drives behavior in the animal. And so we're using this approach really for two main goals, sort of for ongoing work. The first, as I said, is to understand how disruption of the gene RAI1 might lead to changes in the patterns of this kind of activity, essentially to come up with a neural signature of RAI1 dysfunction. And so I'm not going to present the details here tonight, but what I shared with the research panel just a few months ago was that we already have in our first year of funding some really cool results showing very interesting differences between the RAI1 mutant mice and their wild type siblings in many cases, leading us to think already that there are some signatures there that we can pull out, that we can study. And that leads us to the second goal and what is a big part of the second year of funding, which again, we're incredibly thankful to have received. And that's to also start looking at whether or not some of the therapeutic interventions might serve to correct or at least partially ameliorate some of these functional changes that we see. And actually one thing that I didn't mention as well is to also try to link those patterns to specific behavioral outputs in the mouse. And so again, these are our two major goals for the coming year. And we're really excited to continue this work. And so I'll just finish by pointing out we just had our first sort of in-person event as our joint labs just a few weeks ago. We are incredibly grateful to your community, and we're really enthusiastic about trying to give back from the generosity that you've shown with hopefully some important new biological understanding. So thank you.
Oh, you're welcome. And thank you for all the work that you do. We're really excited to work with you for another year. And, we look forward to collaborating with you for many years. And hopefully you'll also be able to look for something like an NIH grant at some point. Do you think that would be a reasonable?
Yes. Yeah. Sorry, I should have mentioned that. Absolutely. But, so most of what and actually this is maybe a good point to make. So most of the work that our labs do is funded by the NIH. These are sort of large grants that support major multi-year research programs. Quite honestly, I mean, the funding that we receive from you all, from the SMS Research Foundation is quite literally an amazing seed grant. This is just work that we would not have been able to start. We would not have done without, the support that you all have given us. And we are well on our way. I mean, to be honest, I mean, with the data that we've already got, we could start to put competitive, applications together. I think, with the second year of funding, we're going to be an amazing space to continue to get NIH level funding. And we expect to continue this for a long time.
Yeah,
but thank you.
So motivating. So we thank you very much. And we look forward to working with you. So have a great evening. And thanks again, Mike.
Thank you.
You're very welcome. So in the interest of time, I'd like to thank all of our researchers for their time and efforts. We really appreciate it. I'd also like to thank our scientific advisory panel, which I didn't do formally, but all of their time is volunteer, and they spend a lot of time looking at projects, deciding on projects, meeting with researchers for quarterly and semi-annual updates. And so I want to thank them for that as well.
And so amazing, incredible research. It blows me away anytime I hear it and to listen to these researchers talking about the work that they're doing and to know that our organization is behind funding it. Of course, none of it would be possible without the help of our donors and our families that support this effort. And of course, this event that went on for the entire month of June,
yes,
our walk, we'd like to take a minute to recognize, and I'm going to go ahead and I think we lost. Oh, here we go. Okay.
We're going to one thing out before we start thanking our teams, because as you said, Missy, our virtual event, I am blown away by how amazing this virtual event has been for us. This is our second one. We did one back in the fall. We had tremendous support for that, and it allowed us to truly become an international fundraising event, which has been wonderful and has allowed our board to work together for one event, which has been great. So thanks everyone for that. We'd also like to take some time to thank our sponsors.
Yes. So our sponsors are, thank you so much to Dewey Insurance Agency, to the Fairweather family, Bob and Carrie, thank you. Priority Lending, thank you. Nye, Sterling, Hale, and Miller, LLP, thank you. Living Special Needs, Title Guarantee, Frater Cangeli Wealth Management, and Gift Baskets R Us. Thank you so much for sponsoring this event tonight and the walk all month long.
Now we'd like to start thanking all of our teams. We had quite a turnout this year. In total, we had 18 teams join us. And on our last event, we had 7. That's a tremendous growth for us in supporters, and we can't thank you enough for that. Last year we had 78 participants. This year we had 118 participants. And last year we had 201 donors, and this year we had 228. We can't thank you all enough. And because of all of that incredible support, we absolutely not only reached our goal, we exceeded our goal. As you can see, we're up to over $68,000, almost at $68,000. And it's incredible. Thanks to everyone who has jumped on board. We really appreciate it. Now on to recognizing our teams.
Yes, let's start with Relay for Research. This is Cole Ianuzzi and friends from Connecticut. Thank you so much for your support.
Our next team, Sienna Steps, the Longman family from Florida.
And we have Jeremy's Journey, the Diamond family out in California. Thank you, Diamonds.
And the Strength for Sydney team, the Ianuzzi family.
Mm-hmm. We have Robin's Super Squad, the Selden family from Michigan. Thank you.
We have Brooklyn's Besties, the Miller family from Arizona. Thank you all so much.
We have Walking with the Sour Patch Kids, adorable name, the Sour family in Texas. Thank you so much.
And Ben's Bunch, the Kingston family from California. Thank you all so much. You did a great job.
Team Jocelyn, the Doring family in New York. Thank you.
Steps of Hope for Valentina, the Perazzi family. Thank you all so much. They're from New Jersey.
We have Team Miller, the Miller family from California. Thank you.
We Against SMS for Amelia, the Quaid family. This is our international family from Germany. They have been tremendously supportive. Thank you for joining us from overseas.
Then we have Stepping It Up for Hunter, the Fegan family from California. Thank you.
I'm going to jump ahead and hopefully the slide will follow us.
You want me to get it? It's
Team Wisconsin, Roll Out the Barrel for Research. Manny Garzinski from Wisconsin. Thank you. The next one is Jeffrey's. Jeffrey's
Try, Dolores Guy, the Guy family from Michigan. Thank you.
Okay, next up, Ava's Angels, the Lewis family from Ontario, Canada. Thank you so much.
And we have Treads for Trey and SMS, the Wampler family from California. Thank you.
Team McKay, the Braun family from Montana. Thank you so much.
And last but certainly not least, Miles for Mitchell, the Morrison family from North Carolina. Thank you so, so much. I'm sorry. We have one more.
Raising funds in honor of Gavin, the Corbett family. Thank you so much. The Corbett family is out in New Orleans, I think, and this is her sister raising money. Thank you so much for that. Well, that wraps up our team support. Now on to our t-shirt. Missy, share with us this beautiful t-shirt we have here.
Yes. So if you were part of the SMS Research Foundation or were aware of the SMS Research Foundation a few years ago, we did a version of this shirt where we include names of children with SMS on the back of the shirt. So we are updating that t-shirt. This is what it's going to look like. There is still some time to get your child's, your loved one's name. This is an example using the names from the last shirt. So if you make a donation of any amount to the event or to the organization, you can also get your SMS loved one's name on the back of the shirt, and then the shirts will be available to order in July.
Beautiful, thank you. So to wrap things up, I'd like to share some final words from one young man who really can't really say it better than the rest of us. We have one last push for you. If you take a moment and hear from Ben Kingston and perhaps follow his advice.
Hi, I'm Ben, and I have Smith-Mcginnis syndrome because of a switch on the 17th chromosome. This month, we are raising money and awareness for Smith-Mcginnis syndrome research. So, if you could please do 17 or something and raise some money so we can learn about it. It would help me and other people with Smith-Mcginnis syndrome if you did. And thank you to everyone who has already donated.
Thank you, Ben. I thought he did a phenomenal job. We'd also like to just take one last chance to thank our sponsors, all of our teams and our donors. Let you know that it's not too late. We've extended the deadline for joining us till July 4th. So it's not too late to do a little 17 of something if you'd like. Also, if you'd like to make a donation, you can go log on to our website. In the chat as well is posted the link to our event. Any other words from you, Missy?
Please consider, making another donation, especially if this research inspires you, moves you. Join us and be a part of it. We are doing incredible things thanks to these amazing researchers. So we love and appreciate all of you and all of your support. The links are in the chat. Thank you so much for joining us tonight.
Good night, everyone.
Have a great night.
Bye-bye.